Hi everyone. In today's session I'm going to be talking to you about how we prepare cells in order to study them. In order to study the individual organelles in detail, we often need to obtain large numbers of these organelles in isolation. This process of preparation of organelles is called cell fractionation, and it's done in two steps. The first step is homogenation, and the second step is ultracentrifugation, and we're going to talk about each one separately. So before we can even start this, we need to place the tissue that we want to study in a cold, buffered solution with exactly the same water potential as the tissue that we're studying. Why does it need to be these three things? Well, it needs to be cold because we essentially want to reduce or stop any enzyme activity that might break down these organelles. So we don't want to cause any damage to our organelles via enzymatic action. It needs to be buffered so that the pH does not fluctuate. We know that any change in pH could alter the structure of any proteins or proteins within the organelles or the organelles themselves, or it could affect the functioning of enzymes within those organelles and therefore inhibit our ability to see how those organelles function in our experiment. And why does it need to be the same water potential? This is because we want to prevent the organelles from bursting or shrinking as a result of osmotic gain or loss of water. So these three things, cold, same water potential, and buffered, are all there to just keep the organelles as similar to how they are in life as possible. So let's talk about the two stages. Homogenation. This is essentially where the cells are broken up by a blender or a homogenizer. We blend the cells and this releases the organelles from the cell, and you get this fluid called a homogenate. This fluid is then filtered to remove any complete cells or large pieces of debris from our tissue. The second stage is ultracentrifugation. So after you have filtered the homogenate, you then put the homogenate into a centrifuge. Centrifuges spin at very, very high speeds, and this creates centrifugal force. So the filtrate is placed in a centrifuge, and first it will be spun at low speeds, at slow speeds. At these slow speeds, the heaviest organelles will fall to the bottom. So the organelles will start to separate according to their density due to the centrifugal force. And the heaviest will go to the bottom of the tube. So in this case, the heaviest is the nucleus. So the nuclei from our tissue will go to the bottom of the tube and they will form a sediment or a pellet. And then we'll get all this fluid at the top, and the fluid at the top will contain all the other organelles in the cytoplasm. So we remove all this fluid at the top, and we call this fluid the supernatant, and we get just the sediment of our nuclei. Then we basically repeat the process. So we transfer the supernatant to another tube, and we spin it at a faster speed than we did before. When we spin it at a faster speed, the next heaviest organelles, which are the mitochondria, get forced to the bottom of the tube. So you have your pellet containing your mitochondria, and then you have the supernatant, the fluid at the top. This fluid at the top is then removed, and we've got our isolated mitochondria, and we continue this process, each time spinning it at the centrifuge at a higher speed, creating greater centrifugal forces, and we separate out the organelles by density. So by the end of it, we'll get all these different organelles in isolation. And we can either then use these organelles to study how they function, or we can look at what they look like. We can look at their structure. You need to know this process. You need to be able to describe the process. And you need to know these key terms. So you need to know the terms supernatant, centrifuge, and pellet. So take a little bit of time to go back over that process and check you're happy with it. Well done.